students. We know that there are various physical methods of the preservation of the food, and temperature holds a very important aspect in that preservation process. Today in this lecture, we are going to discuss other physical methods other than temperature which are used for the preservation of the food in the food industries. The first technique that is used for the preservation of the food is radiation, or we say food irradiation. The process of food irradiation is also called cold pasteurization or electronic pasteurization, because this pasteurization takes into account not the heat but the electromagnetic wavelengths, and this takes place in the absence of the heat, and therefore this is cold pasteurization or electronic pasteurization. If I consider the radiation, there are basically two types of radiation. One is called the non-ionizing radiation and the other one is called the ionizing radiation. In the non-ionizing radiation, we consider the electromagnetic waves which do not ionize the atoms and the molecules present in the food. However, in the other case, we consider the radiations as ionizing, which ionize the atoms and the molecules. In continuation to our non-ionizing radiation, let us study the different wavelengths which are used for the preservation of the food. The first source of non-ionizing radiation is a very common apparatus that we use in our kitchen. Yes, we very well know our microwave oven. The microwave oven produces the waves which are known as microwaves, which is produced with the help of a component present inside the microwave, and that is called magnetron. Now this magnetron which is present inside the microwave produces the waves which are of the frequency between the infrared and the radio frequency, that is from 109 Hz to 1012 Hz. It has got relatively low quantum energy and therefore it causes the production of a thermal energy. Now what is the effect of this microwave on the microorganism that is present inside the food? Now the microwave, when generated inside the oven, produces or it causes the water molecule that is present inside the food to move, and therefore they start moving rapidly back and forth. As a result, a kinetic energy is being created. Now this creation of the kinetic energy causes a rise in the temperature inside the food item, which is passed on from one molecule to the other neighboring molecule, and as a result the food gets heated up. Now this energy which is produced by the microwave by the help of the kinetic energy of the water molecule can kill the microorganism that is present inside the food and therefore helps in the preservation process. Another non-ionizing radiation that is very much used in the food practice or in the preservation of the food is the UV radiation or ultraviolet radiation. If I consider the spectrum of the light, the UV light wavelength ranges from 100 to 400 nanometer. Now this UV light wavelength is being further divided into UVA, which comes in the range of 315 to 400 nanometers, UVB, which is in the range of 280 to 315 nanometer, UVC, which is in the range of 200 to 280 nanometers, and then we have got vacuum UV, which is in the range of 100 to 200 nanometer. This region or the vacuum UV is not at all antibacterial in nature. However, the range from 200 to 300 is called microbicidal because this is the range which is antibacterial in nature, or this frequency is able to kill the bacteria that is growing inside the food item and therefore helps in the preservation of the food. Now the penetration of the UV is not so good if I consider the ionizing radiation. Why? This penetration power is different in different media. For example, in the case of air, the penetration power of the UV is 300 to 500 centimeters. In the case of water, this is 30 centimeter. In the case of glass, this is 0.1 centimeter, and in the case of milk, this is 0.01 centimeter. So the penetration of the UV is not so good, and this keeps on changing when the media changes. Now, what is the effect of UV on the cells or on the microbial community? Let us learn this. When the UV falls on the cell or on the living cell, the DNA gets affected. Now, thymine is a base which is present in the DNA. Whenever this DNA is exposed to the UV light, there is formation of thymine dimers. Here you can see the two thymine molecules combine together and there is formation of a thymine dimer, and this process is called dimerization. Because of the formation of the thymine dimer, now when the uncoiling of the DNA takes place, the dimer is there in its place, and therefore this halts the process of replication and the DNA is not able to replicate. As a result, the cells are not able to survive and ultimately they are killed. The source of the UV light or ultraviolet radiation can be ultraviolet lamps, which are used in the industry where the food or the bulk food items are being exposed and ultimately they are packaged. Also, the UV incubator chamber are small source of the UV lamp where the batches of the food item are kept and are sterilized. A very common UV light can be seen in your household also when you use a water purifier. Yes, the water purifier shows the presence of a blue colored lamp, which is called the germicidal lamp. This is nothing but a UV light source. Here you can see in this picture, this UV or UV lamp or the germicidal lamp is present in this chamber where the water is added or it comes inside this chamber with the help of this inlet. It is kept for some time, which is called the holding time, at which the microorganisms which is present in the water are killed and ultimately the water is taken out with the help of this outlet. So this is the function of UV lamp in a water purifier. Going on to the application of the ultraviolet light for the sterilization and for the preservation, we find that the eggs which are coming out of the poultry should be sterilized or they should be surface sterilized with the help of the UV lamp, where numerous eggs are kept in the trays and they are exposed to the UV lamps, and after this they are surface sterilized. Also in the mineral water purification plant, the water is sterilized or the microorganisms that are present inside the water are being killed with the help of the ultraviolet radiation. Now, whatever the conditions or different processes may be or the applications of ultraviolet light may be, many microorganisms are resistant to this ultraviolet radiation, and this resistance could be shown in this pattern where we find that viruses are very much resistant to the ultraviolet radiation in contrast to the gram-negative bacteria which are more susceptible to the ultraviolet radiation. And the pattern of resistance is like this. Going on to the ionizing radiation, that is the second type of radiation that is used in the food industry for the preservation or for the processing of the food. Ionizing radiation are the high energy waves which ionize the atoms and the molecules along their track in a medium. Now these radiations are basically the alpha, beta, gamma, cosmic rays and X-rays, which are the one which have got a high energy as well as they have got a high penetration power. Now the ionizing radiation are used in the industry for various purposes, but before going in for that, let us study the effect of these ionizing radiation on the food or on the microbial cells which are present inside the food. The main site of damage in the cell is the chromosome, where the hydroxyl radicals cause the single or the double strand break in the DNA molecule. This results in the removal of hydrogen from the deoxyribose sugar. Along with it, there is a further cleavage of the molecule when there is beta elimination of the phosphate group. So this results in the hampering of the DNA molecule, and as a result, the microbes are not able to survive in the food item. Also, an indirect inhibitory effect is seen in the cell constituents by generating the free radicals by the water molecule. Now, the free radicals which are generated from the water molecule, they may either combine with each other or they may combine with the oxygen, and as a result, there is the production of a high oxidizing power. And because of this production of the high oxidizing power, which now act as a powerful oxidizing agent, it damages the cell components. So we find that if there is an absence of water and oxygen in the food, then we have to subject about 2 to 3 times more amount of radiation in order to bring out the same lethality. Because if the water is not there, you will not have the production of the free radical and they will not combine with oxygen, and therefore you will not have the generation of the oxidizing power. Coming on to the application of the ionizing radiation, we find that the food item like pulses, spices, they can be irradiated with the help of this ionizing radiation when we get rid of the microbes which is present inside them. And also fruits, the different type of fruits can also be irradiated with the ionizing radiation. However, there is a pattern of resistance shown by the microbial community against this ionizing radiation, where we find that the viruses again are the one which are more resistant to the ionizing radiation in contrast to the gram-negative bacteria. After radiation, there is another physical method which is very much used for the preservation of the food, and that is use of high pressure. Pascalization is a method where we use a high pressure for the preservation of the food. The high pressure processing unit can extend a product's shelf life and it minimum or with a minimum degradation of the quality of the food. Here you can see a picture of the high hydrostatic pressure processing unit. It means the pressure is applied with the help of a liquid, and therefore this is called the hydrostatic pressure unit. The stirring-flow retort is a giant pressure cooker which is again used by the industry in order to create a high pressure or in order to preserve the food material by subjecting it to a high pressure. This is a large pressure cooker placed in an horizontal position, and here you can see the lid is being opened up and the materials will be packed inside this, and when this is closed, a high amount of pressure is created and therefore it sterilizes the product. Now, if we learn the effect of the high pressure, we find that it has got two different types of effects. One is called the positive effect and the other one is called the negative effect. The positive effect is the one where we find the pressure is given beyond 100 megapascal. However, the negative effect is seen if the pressure is given below 100 megapascal. In the positive effect, we find that deactivation of the enzymes takes place when the tertiary and the quaternary structure of the enzymes are altered. And as a result, the microbes are not able to survive and they will be killed. Most bacteria in their vegetative form are inactivated by 400 to 600 megapascal of pressure, but the spores or the bacterial spores like the endospores are able to resist a pressure of 1000 megapascal. If I consider the negative effect, we find that activation of the enzymes may take place and therefore it saves the enzymes from denaturation. So negative effect is an important aspect of pascalization. Application of pascalization is mainly done in the industry where we have giant Pascal or pressure cookers where the industrial food high pressure processing unit are maintained. Also the isostatic pressure chambers are there in the industry which goes in for the fruit juice preservation or where they get rid of the microbial cells. After pascalization, let us now consider another physical method of food processing and preservation, and that is ultrasound. The ultrasound takes into consideration the range of frequencies which are above the human hearing, that is 16 kilohertz, and they can be propagated in liquid media as an alternating compression. Now, ultrasound can have low intensity or it can have high intensity. When the ultrasound of low intensity is used, it acts as non-destructive analytical method. Here in this case, we can assess the composition and the structure of the food. That means what are the basic constituents that are present inside the food. However, if we use an ultrasound of high intensity, this causes a method which is called the destructive method, which is used for the preservation. Here, it disrupts the physical properties of the microbial cell, that is, the destruction of the tissues takes place. It promotes the chemical reaction. It also is used for the cleaning of the equipments. The high intensity ultrasound also create emulsions in the fruit juices. So in this way, the high intensity ultrasound can be used for the preservation. Now the resistance to the ultrasound is again shown by the microbial cell, where we find that bacterial spores are more resistant in contrast to the gram-positive and the gram-negative bacteria. The application of the ultrasound can be seen in the food processing industry, where we find that a particular food item has to be cut. Now, if this food, here you can see in this picture that this is cut with the help of a mechanical blade which is non-ultrasonic. If the same food is being cut with the help of an ultrasonic blade, it will be free of microbes and the cut will be more uniform. So this ultrasound is used for the processing as well as for the packaging of the food material. The high intensity ultrasound is also used for creating emulsions in the fruit juices. The last physical methods that we are going to learn today is the creation of the anaerobic conditions. Now, anaerobic condition means we get rid of the oxygen. We don't supply oxygen to the microbial community. As a result, the microbes will not multiply, they will not survive, and we will be getting rid of the microorganism that is present in the food item. Now, this anaerobic condition can be achieved by completely filling the container, and the excess amount of unspilled space or the air which is present can be replaced either by carbon dioxide or by nitrogen. Now, this anaerobic condition or the creation of the anaerobic condition is very important in the case of spores. There are many spores, I told you, they can resist a high temperature and high pressure. So, they escape the process of canning. However, when the same thing is subjected to the process of anaerobic condition, they will not survive because there will not be any supply of oxygen and therefore they will not grow. The application of the anaerobic condition is seen when we undergo a process which is called pickling, which is a very good method or a creation of the anaerobic condition where the vegetables or the fruits are being subjected to a brine solution or vinegar. Here you can see that we can have production of numerous pickles or preservation of the vegetables. So, we have learned different physical methods of the preservation of the food. This ranges from temperature, which can be a high temperature, a low temperature. This can be ultrasonic waves, this can be microwaves, ultraviolet radiation, high pressure, which is referred to as pascalization, or it can be creation of the anaerobic condition. All these together shows that a food can be preserved for a much longer time according to the bioburden that is present inside the food. So, the bioburden is an important aspect which or after which the food goes in for the preservation. Thank you.